recording, so I have to record this. It has a high affinity binding site. The binding site's in the high affinity state here, and it's outward facing. But that binds the whatever molecule it is, and it transports it through, and the binding site goes to the low affinity state, drops it off on the inside. Yes?
Are you, just going back to the test, are you going to have a review night or anything?
Would you guys like a review night?
Yeah.
Sure.
Okay, I'll do that. Sunday night. What's that?
Oakham has one at 7.
What's that?
Oh, we're having one for Oakham at 7 p.m.
8:30. How long is one for Oakham?
Fine.
I don't know. We're
just
going
talking
to
about
practice
the
it.
test.
So.
Okay. Well, I'm definitely not doing it at 7 because I got to put my kids to bed at 7. So it's not going to be until after that. So we'll do it Sunday night at 8:30. Okay.
In this room? Might as well be in
Yeah,
the-
I'll be here. Maybe I'll have the test written by then.
Ooh.
Maybe.
Ooh.
Take that one, right?
We could just do a group test.
No. I mean, I can't wish that, but no, unfortunately. Anyhow, so there's a couple of these, a couple different kinds of these transporters. We can have uniporters, symporters, or antiporters. So let's take a look at these. A uniporter is going to transport one molecule in one direction. And remember, this is still diffusion, so this has to be down its electrochemical gradient. You're not going to transport anything through a uniporter down its electrochemical gradient. So some examples of this are carbohydrates often get transported this way, like glucose going into cells goes through GLUT transporters. That is a carrier-mediated diffusion transport. We also have amino acids are also often carried this way as well. In contrast to this, we have symporters, and these are going to carry two or more molecules in the same direction. And these are kind of interesting because one of them is going to go with its, at least one of them is going to go down its electrochemical gradient, but at least one of them is going to go against it. So in this way, we can actually get some things now going against their electrochemical gradient. An example of this might be something like this illustration here. We have outside the cell, we have a lot of these orange dots because all of your cells have lots of orange dots on the outside of them. I don't know if you've noticed that before, but they do. And they have very few red hexagons. So the red hexagon is going against, if it comes in, it's going against this concentration gradient, but the orange circles are going with their concentration gradient. So they all bind, and essentially they do the same thing here, go through at the same time, and they can all go through. Now, because these orange ones are going down their electrochemical concentration gradient, their electrochemical gradient, we actually can pay for this one going up its electrochemical gradient. So an example of this would be the sodium glucose transporter that takes two sodium and transports them across down their electrochemical gradient, and one glucose going against its electrochemical gradient. The sodium going down the electrochemical gradient is going to get you delta G of negative 8.37 kilojoules per mole, but it's going to cost the glucose about 7 kilojoules per mole. So you're still in that negative delta G as you do this. So it's still considered diffusion because it doesn't require in and of itself an input of energy. Also in this are antiporters. They're very similar to symporters, but in this case, we have two or more molecules and they're going in different directions. The concept here is the same, however, where at least one is going down its electrochemical gradient, and at least one is going against this electrochemical gradient. An example of this would be band 2 in erythrocytes, where we transport chlorine in to the cell and bicarbonate out of the cell. Bicarbonate is generally going down its electrochemical gradient, and chlorine is going against it. And then, all three of these. So we've talked about simple diffusion, where molecules can just go straight through the membrane. We've talked about channel-mediated diffusion, where it's just going straight through a channel, but it's not binding to anything. We've talked about carrier-mediated diffusion, where the thing you need to transport is actually being bound to a protein and has to be carried across while being bound to that protein. We also have active transport. And this is one or more molecules that's going to move against its electrochemical gradient, and it's going to require a payment of ATP. So the delta G of just the transport by itself, not considering the ATP hydrolysis, is going to always be positive. We're always going against the electrochemical gradient, so ATP is required to pay for that positive delta G. Now these can be very similar to what we've seen before in the carrier-mediated diffusion, in which we can have uniporters. An example of this is a proton ATPase. It's just moving protons across the membrane, and it's doing this by hydrolyzing ATP and against the electrochemical gradient. We use these a lot for pH regulation in cells, or even pH regulation of the extracellular fluid around the cells. It is also common for us to have antiporters in active transport. One really common example of this is the sodium-potassium ATPase. Whoa, that looks weird. I swear, it doesn't look like that on mine. Looks normal. Okay, hey, it looks completely normal on here. Anyhow, this says, okay, so sodium, potassium, ATP, this one line is just so funky. That's 1/3 of all animal metabolism is actually tied up in a sodium, potassium, ATPase. So if you're sitting here, you're just sitting here doing your thing, you're burning energy, 1/3 of all that energy is just going into running this one molecule. I mean, the whole population of this one type of molecule. Not one molecule, one place in the brain. And we're going to talk actually about why these are so important. These are insanely important throughout the entirety of your body. We're going to talk in a few minutes about why in the world that is. But just know, your sodium, potassium, ATPases, they're everywhere, they're insanely important, and they're super power hungry. What they do is they are going to transport sodium outside the cell, from the inside to the outside. The outside of the cell is sitting at about 140 millimolar. On the inside, you're at about, most cells, this is on average, not every cell, you're sitting at about 12 millimolar. So there's a pretty steep concentration gradient, like a tenfold, more than a tenfold concentration gradient you're pumping the sodium against, not to mention its electrical gradient. And then, also potassium, you're taking potassium from the outside of the cell and pumping it inside. And in this case, again, we have even a greater concentration gradient going that direction. So both of these are very energetically demanding tasks. With each cycle of the sodium potassium ATPase is going to pump 3 sodiums out, 2 potassiums in, and hydrolyze 1 ATP doing it. Okay. So up until this point, we've talked about a lot about membranes, what goes into them, the lipids that go into them, the type of functions that we can do with them, and the type of transport that can happen across them. So for the last little bit, this next section I'm going to go into is the last bit for the test. It's going to pull it all together by talking about one of the more complicated functions of a cell membrane, which is to maintain membrane potentials. essentially all cells in your body have some sort of membrane potential. And all we mean by a membrane potential is that we have a separation of charges across the membrane. We've taken charges and we've segregated them on different sides of the membrane. Your neurons and other electrically active cells are most commonly known for this, but essentially all of your cells have some sort of membrane potential. And we call it potential because this separation of charges that we build up has the potential to do work. We can do things. Once we separate those charges on either side, we can then harness that to do things with that. So what kind of things might we want to power with this? There's a couple examples of this. So what's the purpose of a membrane potential? One of the things that we could do is to power membrane-bound enzymes. For instance, carrier-mediated diffusion. Remember we were talking about the symporters and the antiporters. One of those molecules is moving down this electrochemical gradient. It can do that because we built up a membrane potential, and actually the electrical gradient, or that membrane potential that we built up across that membrane, is doing most of the heavy lifting in most of the symporting and antiporting that you have going on in your body. It's actually this electrical potential that's driving that carrier-mediated diffusion in which we have one thing moving against this gradient. Another example of something that we could use for this is, we're going to come back and talk about this later in the quarter, but mitochondrial ATP production happens largely because of electrical potential or a membrane potential that we built up on a mitochondrial membrane. Now this case is slightly different because it's on a membrane-bound organelle and not on the cell membrane, but the principle is essentially exactly the same. The other purpose of a membrane potential could be that we want to have electrical conduction within this cell in some way. And so in order to do that, we need to build up a membrane potential first in order to enable that electrical conduction. This is something that happens most commonly in neurons and muscle cells, but actually happens all over the place, including like your liver cells conduct electrical potentials across to communicate with each other and few other strange places you might not have thought about. Okay. So let's talk about how basically this happens. Let's talk about this very generally at first. Let's imagine we have a cell membrane here in the middle, and we have some charges. We have some charges on either side. We have some plus charges and some minus charges, and we could count up the plus charges on this side. We have 1, 2, 3, 4, 5. And we have 1, 2, 3, 4, 5 minus charges, and that's equal on both sides. Right now there's no potential, there's no segregation of charges. But let's imagine just for a second that we could drag two of those from the right-hand side to the left-hand side. And so what happens now is if we look at this, we have 1, 2, 3, 4, 5, 6, 7 charges, plus charges over here, and 1, 2, 3, 4, 5 on this side. So we have an excess on this side of plus 2 charge. Whatever units, we'll come back to units in just a minute. And on this side, we have 1, 2, 3, 4, 5 minus charges, but only 1, 2, 3 plus charges. So we have an excess of minus 2 on this side. Let's just imagine for a second that this is the extracellular fluid and this is the intracellular fluid. If we look at the difference across here, we actually have a difference of minus 4 charges. Just using as a convention the charge excess on the intracellular side as a thing, as the convention. We go with the excess on the intracellular side. So these charges really aren't actual pluses and minuses in your cell. I know that you probably thought they were literally plus and minuses, but they aren't. And what will tend to happen is that the excess charges on either side will tend to aggregate near the cell membranes. And so, in reality, you have far more, on either the extracellular or intracellular side, you have far more charges than really need to be used to set up a membrane potential, those excess ones are going to aggregate themselves on the inside of the membrane. So those charges aren't just, again, random pluses and minuses being floating around. They are, in actual, being carried by ions, which you probably intuited before this point. Things like sodium and potassium. The primary ions involved, the positive primary ions involved in this are sodium and potassium. That's primarily what we have carrying positive charges. On the other hand, the negative charges tend to be carried by either large negatively charged proteins, and those tend to be on the intracellular side, or also chlorine anions. And one thing to keep in mind as we go throughout this, it actually takes far fewer molecules to set up an electrical gradient than what it would take to set up an equivalent chemical gradient, to set up a similar force in the chemical gradient. And by far fewer, I mean like many, many, many orders of magnitude. In other words, in most times when we set up an electrical gradient or a membrane potential on a cell, we cannot detect the change in concentration gradient across that cell membrane that caused that. It is an undetectable change in the concentration gradient. Let's talk about the measurement of these potentials. By convention, again, just to reiterate, we're using the excess charge on the inside of the membrane when we're describing how large or how small that potential is. And we usually measure it in millivolts. So for example, if you have a membrane potential of negative 50 millivolts, this means that you have 50 millivolts more negative charge on the inside of the cell than you do on the outside of the cell. You have an excess of 50 millivolts on the inside, more negative. So let's put this a little bit into context. Here's some various resting potentials of various cells in your body. Starting with some of the highest skeletal muscles have about negative 95 millivolt electrical potential, all the way down to things like erythrocytes have about -10, chondrocytes have about -8. One really important one is neurons. Neurons have about a negative 70 millivolt. I really don't care if you know the rest of these, but neurons, you're going to have to remember that they're about negative 70 millivolts because that's going to come up again. And neurons are really what we discovered. Like, we figured out what was going on with membrane potentials by studying neurons. They were the first cells that we really identified that had membrane potentials. Partially because we knew they were electrically active. We're trying to figure out what it was. As a little aside, does anyone know what animal we first figured out how neurons worked on and first discovered and first started measuring neurons?
Squid.
Squid! Cephalopods. Do you know why it was in squid? Yeah, so I had little things all over the place. Yeah, they have a giant axon in terms of, it's not so much, it's not longer actually than injuries. You actually have longer axons, but it's huge and thick. And you mentioned myelin sheath. It's not because they have a big myelin sheath. It's actually because they have no myelin sheath. It's that way because they have no myelin sheath. And so, like everything, like the whole foundation of neurophysiology we know of because of squid. And if squid didn't exist, it would have set us back 20 years in neurophysiology. So, you should be glad. What's that? Yeah, squids are super important. So studying the physiology of cephalopods is a really important endeavor. If any of you want to do research in my life, just hit me up now. Anyhow, so neurons, they are kind of the quintessential membrane potential. So However, all of these kind of pales in comparison to the membrane potential on the inner membrane of the mitochondria is even twice as large as the neuron. And it's not even like a whole cell. It's negative 60 millivolts across the inner membrane of the mitochondria. Again, this is something we're going to come back to a bit later, but it's absolutely ginormous. And yeah, it's just a little thing. So let's talk about this specific. Oh, do you have a question?
Quick question.
Yeah.
If the electrical gradient is set up with all these other kind of atoms, what's the difference between electrical and chemical?
Let's talk about that. That's a really good question. In short, let me just give you a short answer to that. You set it up with those, but the electrical gradient, you only have to move very small amounts of these ions across. And so, by definition, you will change to some degree the concentration gradient across there. But it's so minuscule compared to the electrical gradient that gets changed that the difference is that just the electrical gradient is going to be much larger in magnitude.
So every electrical gradient is a chemical gradient?
Yes,
Okay.
to some degree. Now, that's not strictly true in all cases, but in most of biology, that is true. There are exceptions to this, but that's generally true. Yeah, it's a type of chemical gradient. But the problem is that it actually is realized in a very different way. So there's a good reason for calling it a completely different thing, because the interaction between the molecules, because you have an electrical charge on them, is very different just than the interactions you have just because you're there. Like a neutral molecule and another neutral molecule, and they just, because of concentration.
So you're saying the cell is a lot more sensitive to the electrical?
It's not so much the cell itself, it's just physics. Okay, so let's talk about specifically, how do we create an electrical potential? So this hopefully also gets at your point in a little bit more detail. How do we set up an electrical potential? And it honestly is slightly different in all cells. Like when we look at how it gets set up in the mitochondria, it's actually radically different than this. Most cells, however, do something that's slightly similar to this. And this is specifically how it happens in a neuron. And so what you'll see, if you just kind of looked at a neuron, what we have here is we have an extracellular fluid that's rich in sodium and relatively poor in potassium. We have it said extracellular, right? Not intracellular? Extracellular. We have an intracellular fluid that is very rich in potassium, but fairly depauperate in sodium, but also has these fairly large anions, these large proteins bumping around in there. And so we have more negative charge here. We also have in the membranes, we have our sodium potassium ATPases here, and they're doing their thing, pumping 3 sodiums out and a couple of potassiums in, 3 and 2 each time. And then we also have a really funny thing. We have sodium leak channels and potassium leak channels in there. These are just channel-mediated diffusion. They're just channels that have no gates, and they just let all of that stuff that the sodium potassium ATPase did, they just let them leak back the other way. And so what you get is you get some sodium leaking back into the cell, you get some potassium leaking back out of the cell, and there's one really key important thing here. There are tons more of the potassium leak channels than there are the sodium leak channels. There's somewhere in the neighborhood of 30 times as many of these potassium leak channels than the sodium leak channels. And that becomes the key to this entire thing. The sodium potassium ATPase here is pumping charges back and forth, and it's actually pumping more positive charges out than it's pumping positive charges in, even though it's just shuffling around positive charges. You might think, hey, that sodium potassium ATPase by itself can give us an electrical difference across there, and it can to some degree. But it only accounts for, the actual work of this only accounts for about a negative 3 millivolts of the electrical potential in a neuron. So a tiny bit, like 1/20th of the entire electrical potential is coming from the direct action of this. Mostly what the sodium, potassium, and ATPases are doing are setting up the large concentration gradients that we see different between the sodiums out here and the potassiums in here. So the sodium ATPase is driving those differences, those concentration differences. Okay, so we're starting to set this up. Let's just remind ourselves of the different forces that are going to be acting on these ions. We have concentration gradients, and the concentration gradients essentially never get disturbed in normal cell function. Unless you have something really catastrophic happen, they just kind of are set. Because there's essentially always a surplus of ions in the extracellular and the intracellular fluid. Remember how large we're thinking like a cell is compared to the components of the cell, or even smaller cells. It's just a huge thing compared to the individual components of the membrane. And all of our charges that set up the electrical potential are crowding right behind the membrane, like right up next to the membrane. And behind that, there's this huge well of essentially vast quantities of all of those ions just further in the cytoplasm or further on the extracellular fluid. If you lose some of those ions from that membrane, it's really easy to replace them from just back behind there. And so the concentration gradients never really get disturbed all that easily. However, no, let's just play this out. So just remember, relatively few, and then the other part here is, it doesn't take that many ions changing sites to change the electrical potential across the membrane, especially compared to what it takes to change the concentration gradient. So we have this huge surplus, and it takes tiny amounts to change the electrical potential. So for that reason, we can kind of think of the concentration gradients as being a fixed thing across those membranes. It's being maintained by the sodium, potassium, ATPase. If you shut them all down, eventually you would deplete it. But in the course of setting up a potential, it would take a long time. Sodium concentration is essentially always greater in the extracellular fluid, potassium in the intracellular fluid. The other forces, again, just to refresh us, that are acting on these ions is the electrical gradients, where we have positive charges that are going to tend to be attracted to negative ions, and they're going to tend to be repulsed from other positive ions or other positive charges. This is also true vice versa. Negative ions will be attracted to positive ions, but repulsed by other negative ions. And so this helps us set up these electrical gradients. Okay. So if we take out the sodium, potassium, ATPase out of our little model there, and just say, okay, so it's been setting up the electrical gradients, or the concentration gradients. Let's think now just individually about those leak channels, one at a time, and try to figure out what they're doing. Okay, so we have a sodium leak or a potassium leak channel here, and it's just going to passively let potassium leak through here. So let's start thinking about our individual forces acting on there. Concentration gradients. Where does the concentration gradient want to push the potassium? Out, yeah. So that's going to want to start pushing it out this way. Which way does the electrical gradient want to push it? Less certain. In, yes. So the electrical gradient wants to push it in. So concentration gradient in, and again, this is our concentration difference, about 5 millimolar on the inside, 140 millimolar, or 5 millimolar on the outside, 140 on the inside. And then we have our electrical gradient wanting to push it in. It actually turns out in the normal state of things, in a normal membrane potential, if we've already had a normal potential set up, the greater of these is a concentration gradient. And so it's going to tend to push sodium ions out, or potassium ions back out. Okay, now let's just imagine for a second. Let's imagine you just had a cell full of potassium leak channels. We started with something, and we just start letting the potassium just leak out. Would it continue to leak out until it got to equal concentration gradients on either side? No, why not?
That's why the electrical would be going.
Yeah, because then you have the electrical gradient pushing back. And so you continue to have the electrical gradient. So it will not go to even concentrations if you just let it go. Would it go to electrical even? Electrical even on both sides? Why do you say no? Yeah, you have the concentration gradient pushing on it. So you can never, it will never go to equilibrium either in electrical gradient or concentration gradient because they are both simultaneously working on it. And so since you have them both in opposition, they'll eventually balance each other out, where they both have the same force, and you have no net movement of potassium either way. You'll still have movement back and forth, but it'll be equal in both directions, and it'll just stabilize there. This is what is known as the equilibrium potential of one particular ion. And the equilibrium potential of potassium by itself in most cells that have this concentration gradient, is negative 90 millivolts. So we would continue just to go back and forth until we reach an electrical gradient of negative 90 millivolts. And again, remember, we can take the concentration as essentially static because we just have such a surplus of concentration of each of these to draw from, and it takes very tiny amounts of these ions moving back and forth to actually set up the potential. So that in truth isn't going to change. Now, if there's no electrical potential, we just let it go for forever. Yes, it would, but in biological terms, we can just think about it as being constant. Okay, so let's go to the other side. So now we have a sodium leak channel. We also have these sodium leak channels. And so which way is the concentration gradient pushing the sodium? In, yeah. So it's going to tend, well, yeah, we have in the electrical, which way is it going? In, okay. The electrical is pushing in, and the concentration gradient is pushing in. And so, and again, just to remind you what that concentration gradient is 140 millimoles to 12 millimolars. Okay, so it's both pushing in the same direction, like we have like combined. Before we had opposing with potassium, but in this case they're both going the same direction. Okay, so does anyone want to take any guesses about what you think the equilibrium potential of sodium would be? Let me ask you this. Do you think it would be negative or positive? Positive. Why? Positive. They are positive charges, but so are the potassium. Yeah, they're both working in the same direction. Yeah, so essentially you have to go this way enough until the electrical gradient flips and starts opposing the concentration gradient. It's going to continue to flood in here until it completely flips. And to do that, you have to be positive in here. It will not flip until it's negative, or until it's positive here and not negative, net negative. Does that make sense? Okay. Till then, it'll be a less of an electrical gradient, but it'll still be electrical gradient until you're positive in here. Okay, so how about this though? In terms of magnitude, do you think the equilibrium potential, so now I'm saying the absolute value, I don't care if it's plus or minus, do you think the equilibrium potential of sodium is greater or less than that of potassium? Why do you say less?
Well, this is not really logical. I mean, it is logical, but if the neuron is supposed to be 7 millimoles, and the sodium or the potassium is maybe 90, so the difference is 20, so it's less.
That's really good. You're going backwards to get to the right answer, and that is a correct consequence. But it's not the reason. You're going from the consequence back to the thing, which is you're absolutely right. But does anyone know what the reason is? Yeah, Nick.
Aren't there a lot less sodium leak channels, and then you have the active transport where you're pumping in?
You were right. You're absolutely right. There are less sodium leak channels. But with equilibrium potential, we're imagining there's only sodium leak channels, and we just let them go until it's equilibrated. So actually, concentration, how many of those channels we have, doesn't matter in this case. There are, but the main thing is that's just going to make the rush happen faster. That your electrochemical gradient is going to be higher, but it doesn't tell you about the end potential. Okay, so electrical potential is going to go until it matches what? It matches but opposes the concentration gradient. Okay, what was the concentration gradient in potassium? It's 140 to 5. And what is it in sodium? 140 to 12. Which one has a greater concentration gradient? Potassium. Okay, so they're going to equal out. Potassium has the larger concentration gradient difference. And so the electrical potential, the electrical gradient that will eventually even it out, has to be greater than what it is in sodium because you have a smaller difference here. It's not a lot smaller, but it's smaller. So it actually turns out that, okay, so we're pumping those through, and the difference, the equilibrium potential of sodium in most cells with this concentration gradient is plus 60 millivolts. And so therein lies the key. You have two equilibrium potentials that are trying to get, those two leak channels have two equilibrium potentials they're trying to reach. You have on one end, you have sodium at plus 60 millivolts. On the other end, you have, again, this is the sodium equilibrium potential. We have the potassium equilibrium potential down here at negative 70 millivolts, or excuse me, negative 90 millivolts. And just like you were saying, Caleb, because we have so much more, if we had equal, it'd probably end up being here, about halfway in between, where it is like negative, 10, 20 or something like that. But because we have so many more potassium channels, or potassium leak channels, is going to end up way over here, closer to this, at 70 millivolts, as the rate of the sodium and the potassium equilibrate to each other. Yes, that's negative, sorry. Negative 70 millivolts. So that's how it is in a neuron. Now, you start changing a few things. You change the concentration gradients of these across here. You change the equilibrium potential of both sodium and potassium. If you start messing with these concentration gradients, because you change the concentration gradient that the electrical gradient has to oppose. If you change the relative ratios of the sodium and the potassium leak channels, you can also then change it anywhere between here, depending on what it is. And so this is one of the most common ways in which cells set up their electrical potentials. We're shuffling around sodium and potassium inside and out of the cell, and the really counterintuitive thing is that it's just the passive leak of them trying to go back down their concentration gradients that ends up setting up the electrical potential. So, a recap. We have sodium, potassium, ATPases pumping out sodium and in potassium. That sets up a large concentration gradients of both of those. Then we have a few sodium leaking back in and a lot of potassium leaking back out, and that sets up an electrical potential or a membrane potential across this membrane of about negative 70 millivolts.
So the sole purpose of the to set a concentration.
That's the primary purpose. It still gives you about negative 3 millivolts, so it gives you this tiny little amount as well. And if you actually do the math, just looking at this equilibrium potential and this equilibrium potential and about the 130 doesn't quite get you right here. There's a few other things that go into that. But this is the vast majority of what's setting that up. But you do have other things like the sodium potassium ATPase that's kicking in a little bit as well that adds into that. And then you actually have other ion transporters. Like, we're also dissipating these potentials as well, because we're still using them for like SIM porters, We're letting sodium back in to carry glucose across and other things like that. So there's other ways that sodium and potassium are getting across the membrane rather than just the leak channels as well. So if you try to just look at this and do the math to say, okay, if we have 1/30th the rate here of this, do we come out to this? No, not exactly. But those different things are like, we're talking within like each one of them 1 to 2 to 3 millivolts each, not a lot. Okay, just summarizing that. A membrane potential is a separation of charges. Any potential is a separation of charges. A membrane potential is a separation of charges across a membrane. And that we have a sodium-potassium ATPase that's going to set up these concentration gradients. It's doing that using energy. We're using ATP to actually drive that concentration gradient. And we also get about -3 millivolts of membrane potential from that. So we get a little kick in on that. This is happening in essentially every cell of your body. And this is why sodium-potassium ATPases are so power hungry over the course of like your entire body, is because we are setting up that membrane potential. This is also why your brain is especially a super power hungry organ, because we are running tons of these in every neuron. Not only do they set up a proportionally larger membrane potential, but they dissipate and have to reform that membrane potential a ton compared to your other membrane, or most of your other cells. Take animal physiology, we talk more about that. Essentially, you open up sodium channels and you just let all the sodium, not all of it, but sodium flow back. So we actually go to that positive. We actually will flow back this way. And you go from having 1/30th of the conductance of sodium to like 100 to 1,000-fold the conductance of sodium. It all of a sudden rushes this way. This equilibrium rushes this way and gets all the way up to about +10. Before then, potassium channels open and come back this way. Anyway, that's a little aside. You don't need to memorize that. And as the passive leaking, that ends up actually doing most of the work to set up the membrane potential. We have sodium leaking back into the cell, potassium leaking back out of the cell, and because we have approximately 30 times more potassium channels, that is how our actual equilibrium potential gets set up. Or our membrane potential gets set up.